This is the second part of antibody engineering. Okay, and we will start with engineered monoclonal antibodies. So, by using recombinant DNA technology, it is now possible to design and construct genes that encode monoclonal immunoglobulin molecules in which the variable regions of desired specificity come from one species and the constant regions from different species. Okay, and say for example, if we use uh, a region from the human and another region of the antibody from, like, say for example, mice and rabbit, then it will be called humanized chimeric antibodies. Okay, so what the benefit of this technology? So for example, by using these techniques, we can produce different type of antibodies which are specific for different antigens. Say for example, we want to produce therapeutic human antibodies. Okay, so say as we want to use the antibody to treat human disease, so what we can do, we can take the FC portion of the antibody. The FC portion bind with the cells. So we want to use this antibody to treat human disease. So we have to take the FC portion from the human. Okay, and for the variable portion, it recognize and bind with the antigen. Okay, so we can take the variable portions from mice. So why for mice or rabbits? So like we can design mice and rabbits in a way that mice and rabbit will uh, express antibodies, that is the variable portions specific for different antigens. Okay, so what we will do, we will take those variable regions from the, like, mouse or rabbit, which are specific for different antigens, and will make a chimeric, humanized chimeric antibodies which has the FC portion from the human. Okay, so that by using this way we can produce different monoclonal antibodies specific for different antigens, and we can use these monoclonal antibodies as therapeutic agent, that means to treat human diseases. Okay, okay. Engineered antibodies have other applications too, and one application is to treat cancer. So monoclonal antibody can be designed to treat cancer. One approach is producing immunotoxins. Okay, so immunotoxins, they are composed of tumor specific engineered antibodies and coupled with lethal toxins. Okay, and they can be used to kill tumor cells. Okay, so in these immunotoxins, they can be, for their preparation, different toxins are used, such as ricin, shigella, and diphtheria toxins. Okay, so this type of toxins, they usually have two part. One, they have a toxin chain, and another is a binding component. So what is done, the binding component of these toxins are replaced with a monoclonal antibody which is specific for particular tumor cell antigen. Okay, so so if we look at this photo, it will be easier for us to understand. Okay, I'll explain the figure. So first, please look at this left side of the figure. Okay, so this left side, it shows the regular mechanism of the toxin. How they work. Say for example, diphtheria toxin. Okay, so diphtheria toxin has one portion, this portion, which bind with the cell that they infect, and it has another portion which is actually the toxin, which do the damage to the cell. So by this binding, when diphtheria toxin it's infect a cell, by this binding portion it binds with the cell and then get internalized by endocytosis, and once insert the toxin portion, it get activated and it inhibits the EF2. EF2 is a factors. EF2 factors, they are responsible for synthesizing different proteins. Okay, so as the protein synthesis is inhibited by diphtheria toxin, the cells can no longer replicate. That means they can no longer multiply, and eventually they die. So now, if we want to produce immunotoxins using the diphtheria toxin, what we have to do? we have to replace this binding portion of the diphtheria toxin using monoclonal antibody. So what's the benefit? Say for example, if we can identify particular antigen for cancer cell, then we can design monoclonal antibody for that antigen. Okay, so for example, this one is that monoclonal antibody. I mean, this monoclonal antibody, it recognize and bind with particular antigen of certain tumor cells. Okay, so we'll bind, we'll add this monoclonal antibody, tumor specific monoclonal antibody, with the toxin portion. Okay, so now if we use this as a therapeutic agent, so what will happen? So as this monoclonal antibody, it recognizes particular antigen of particular tumor cells, they will bind with that tumor cells, and again they will get internalized through endocytosis by that tumor cell. Okay, and once inside, this toxin part of will they get activated, and they will inhibit the EF2 factors of that cell. That means they will inhibit the growth or proliferation of that cell. So as their tumor cell, so eventually that as those tumor cells cannot no longer multiply, they will eventually die. Okay, so by this way, using immunotoxin, we can treat cancer. That means we can kill the cancer or tumor cells. Okay. Okay, now the last topic of this lecture. In the last topic, I'll talk about phase display or fast display. Okay, so what is fast display and what is its benefit? Okay, so basically by using this technology, we can make a library for different antibodies, and once we can make a library of different antibodies, we can use this library to find out therapeutic antibody against antigen. That means against different disease like pathogenic disease, okay, or cancer, etc. Okay, so what is the basic of fast display? So what is fast? So bacteriophage. Okay, the virus which kill the bacteria. Okay, so by using recombinant DNA technology, we can display the antibody by the phage. Okay, so what actually we do? Say we know the gene of the phage. Okay, so what we have to do? We have to insert our antibody gene into the gene of the phage. Okay, then if as we have changed the gene of the phage, which has the antibody, gene of the antibody, so when the phage will be formed or develop, they will express this gene product. That means they will express the antibody into their body. So in this kind of sense, in during the phage display, basically we choose the coat protein of the bacteriophage to express the antibody. So that means the changes is done in a way, that basically the changes are done in the coat protein, so that after the full bacteriophage is formed, it will express the antibody into their coat portion. Okay, into their protein coat portion. Okay, so by this way we can express different antibodies into different bacteriophages. Okay, so then that means we will have different phases which express different which express different antibodies into their coat protein. That means we will get a library of bacteriophage. Okay, so what is the use of bacteriophage actually? Okay, so as in this library, different bacteriophage, they are expressing different antibody into their coat. So for example, we can use this library to screen out or find out a antibody which can treat our targeted disease. Okay, so for example, we got an infection, okay, and we find out the antigen for that infectious agent, and we want to find a antibody which will bind with that antigen and neutralize it. Okay, so how we can do it? By using the phage library. Okay, so as we have the phage display library, so what we'll do, first we have to isolate the antigen from that infectious particle and then immobilize them into something. Okay, so once we have immobilized the antigen into a surface, then we have to apply this antibody presenting or antibody displaying phases onto that immobilized antigen. Okay, so if we have that antibody, okay, so when we'll apply the antibody displaying phases onto the immobilized antigen, so if there is any antigen specific antibody in our library, so that phase, that antibody displaying phase will bind with the target, will bind with that antigen. Okay, so then we'll just do some washing, so all the unbound phases will be removed from there. Only that phase which express the specific antibody which react with that specific antigen will be bound there. Okay, so then, so what that mean, we got the antibody which can neutralize the antigen. So then from that phase we'll collect that antibody. So, and we already know that what gene that phase has. Okay, so from that gene, by using different expression system like the bacterial expression system or the animal mammalian expression system, we can produce our antibody in bulk amount and can use those antibody to treat that particular infectious agent. Okay, okay, so that's all. Thank you for your patience.